The overall goal of this protocol is to clone multiple guide RNAs into one CRISPR-Concatemer vector and to achieve highly efficient electroporation in mouse intestinal organoids in order to obtain simultaneous knockout of multiple genes.
This method can greatly improve the efficiency of loss-of-function studies by making it possible to overcome the potential effect of parallel compensation. The main advantage of our CRISPR-Concatemer strategy are the convenience of a single cloning step and the possibility of performing simultaneous knockout of up to 4 different genes. Demonstrating the procedure will be done by Alessandra Merenda, a PhD student from my laboratory.
The cloning of guide RNAs, or gRNAs, into the CRISPR-Concatemer vector begins with a single reaction to anneal the top and bottom strands for each gRNA oligonucleotide and to phosphorylate their ends. Prepare the reaction mixture on ice. For 3 concatemers, use 3 microliters of top strand gRNAs, 3 microliters of bottom strand gRNAs, 2 microliters of T4 DNA ligase buffer, 1 microliter of T4 polynucleotide kinase, and water up to a total volume of 20 microliters. Mix well by pipetting and run in a thermocycler using the following settings: 37 degrees Celsius for 30 minutes, 95 degrees Celsius for 5 minutes, ramp down to 25 degrees Celsius at 0.3 degrees Celsius per minute, and hold at 4 degrees Celsius. The next step is the BBS1 shuffling reaction, in which the pre-annealed gRNA oligonucleotides are incorporated into the appropriate position of the concatemer vector. Dilute the reaction mixture 1 to 100 in DNase RNA.